Good day, one and all. My name is Ramnarain. I am a PhD candidate in medical microbiology at the University of KwaZulu-Natal in Durban, South Africa. It gives me great pleasure to present my master's research at the 45th Global Congress on Infectious Diseases. The title of my research poster is on genotyping of Chlamydia trachomatis from vaginal swabs by restriction analysis of the outer membrane protein gene. This study was conducted in South Africa at the School of Clinical Medicine Laboratory, based at the Nelson R. Mandela School of Medicine at the University of KwaZulu-Natal. In both industrialized and developing countries, reproductive tract infections are one of the most important public health challenges. Chlamydia trachomatis, i.e. C. trachomatis, is a gram-negative obligate intracellular bacterium that can replicate only within a host cell. It is the most common cause of bacterial sexually transmitted infections in young women globally. Up to 80% of C. trachomatis infections in women are asymptomatic. Infection with C. trachomatis is becoming more common throughout the world. According to the World Health Organization reports, STIs have risen globally in recent years, with C. trachomatis being one of the most common bacterial STI pathogens, producing an estimated 106 million new infections each year. Infertility, persistent pelvic pain, ectopic pregnancy, early rupture of membranes, spontaneous preterm abortion, and perinatal death are all problems of C. trachomatis colonization in the reproductive tract of pregnant women. Genotyping is a technique used to detect minor genetic differences that can contribute to significant phenotypic changes, such as pathological changes, that underpin disease. Owing to its high resolution, genotyping allows the differentiation of bacterial strains based on their genetic content. Recently, this has become frequently employed for bacterial strain typing. A grouping of bacteria or viruses based on cell surface antigens is known as a serovar. Serovars allow organisms to be classified down to the subspecies level, which is crucial in epidemiology. C. trachomatis is divided into the following 15 serovars: A, B, BA, C, D, E, F, G, H, I, J, K, L1, L2, and L3. Based on the structure of the outer membrane protein OMP1 gene. Currently, there is limited data on the serovars of C. trachomatis detected in pregnant women in South Africa. This study aimed to investigate the prevalence and serovars of C. trachomatis in a population of South African pregnant women. The objectives of the study were to identify circulating serovars of C. trachomatis by restriction analysis, to determine the prevalence of C. trachomatis in the study population by a nucleic acid amplification test, and to link the different serovars with clinical factors. Figure 1 shows a human pap smear showing eukaryotic cells infected with C. trachomatis at 500 times magnification. This study will form the basis of subsequent research on the genetic diversity of C. trachomatis in South African pregnant women. This study was approved by the Biomedical Research Ethics Committee of the University of KwaZulu-Natal. This was a cross-sectional study which included 385 human immunodeficiency virus, i.e. HIV-positive pregnant women, recruited from the antenatal clinic at the King Edward Hospital in Durban, South Africa. Enrolled women provided socio-demographic, behavioral, clinical data, and self-collected vaginal swabs for the detection of vaginal infections. The vaginal swab samples were analyzed for the presence of C. trachomatis. Deoxyribonucleic acid, i.e. DNA, was extracted from the vaginal swabs using the PureLink microbiome purification kits. C. trachomatis was detected using commercial primers and probes, which targets the gene encoding the translocated actin-recruiting phosphoprotein of C. trachomatis. Molecular genotyping of C. trachomatis positive samples was performed by an OMP1 semi-nested polymerase chain reaction assay, followed by restriction fragment length polymorphism analysis. The first product of 1033 base pairs was amplified using the following paired primers, 01A and 02A. PCR was performed with the following cycling conditions: initial denaturation at 94 degrees Celsius for 7 minutes, followed by 40 cycles of denaturation at 95 degrees Celsius for 1 minute, annealing at 45 degrees Celsius for 3 minutes, and extension at 72 degrees Celsius for 3 minutes. An additional 7-minute extension at 72 degrees Celsius was performed at the end of the 40 cycles. Following the first round PCR, 1 microliter of the first round PCR product was used for the semi-nested PCR, amplifying a 978 base pair fragment. The second PCR round was performed with the same reagents and conditions as the first round with the following primers. The reverse primer was 02A from the first round, and the forward primer was PCTM3. After the PCR step, the amplified product was digested with either AlloI, DDEI, or HiNEF-I restriction enzymes and visualized after electrophoresis on a 2% agarose gel. C. trachomatis serovi identification was made by analysis of the specific restriction pattern. Associations between categorical variables were assessed using chi-square tests. All statistical analyses were conducted using RStudio version 3.6.3. All p-values were considered significant when they were less than 0.05. The actin-recruiting phosphoprotein of C. trachomatis was detected in 47 out of 385 swab samples using the Applied Biosystems TACMan assays. The prevalence of C. trachomatis in the study population was 12.2%. Of the 47 C. trachomatis positive samples, 43 had produced positive amplicons for the OMP1 gene. Low levels of education, being unemployed, not being married, not cohabiting, having first sex at a young age, having numerous lifetime partners, not knowing whether their partner has other partners, not using condoms, not showing symptoms of STIs, and not having received STI treatment in the past were factors significantly associated with C. trachomatis infection. Figure 2 shows the AlloI RFLP pattern of the digested OMP1 gene amplicon resolved on a 2% agarose gel. M represents the 100 base pair DNA molecular ladder, showing 100 base pairs, 500 base pairs, 1000 base pairs, and 1500 base pairs, and banding profiles for a subset of 19 out of the 43 clinical samples that generated positive amplicons. Size fragments of 207 base pairs and 256 base pairs were observed for sample CT22. Sample CT21 was the only positive amplicon that showed no restriction banding profile for AlloI. A total of 42 out of 43 samples were assigned serovars. 5 different serovars were detected due to combining the banding patterns across the 3 enzyme profiles. The serovars identified were serovars D, E, F, G, and I. The prevalence of the individual serovars were as follows. Serovar E, was detected in 20 out of 43 samples, 46.5%, followed by serovar F, 9 out of 43 samples, 20.9%, serovar G, 6 out of 43 samples, 14%, serovar D, 5 out of 43 samples, 11.6%, and lastly, serovar I, which was detected in 2 out of 43 samples, 4.7%. Table 1 shows the serovar's linked clinical factors of infection. This study showed that genotyping could be achieved from uncultured vaginal swabs. The observed diversity of serovars and the different clinical factors reported within specific populations can be challenging for future vaccine design and development for chlamydia. However, many of the South African serovars detected correlated with serovars found in studies conducted worldwide. This holds some promise that there may be conserved C. trachomatis strains from different geographical regions and could lend some hope for future vaccine design and diagnostic studies focused on the entire C. trachomatis population. To my knowledge, this is the first South African study to conduct genotyping of Chlamydia trachomatis from vaginal swabs by restriction analysis of the outer membrane protein gene. Future studies emanating from the study would be to investigate the association of the serovars with patterns of drug susceptibility in this pathogen. In addition, it would be useful to compare the distribution of the serovars in pregnant and non-pregnant populations, as well as to further investigate the serovars in connection with the presence or absence of clinical symptoms, possibly by investigation of virulence genes in these specific serovars. I gratefully acknowledge the woman from the King Edward Hospital Antenatal Clinic who participated in this study, and the School of Clinical Medicine Research Laboratory at the Nelson R. Mandela School of Medicine at the University of KwaZulu-Natal, where the study was conducted. This study was funded by the National Research Foundation and the College of Health Sciences at the University of KwaZulu-Natal. These are a few of the references cited in my research paper. Thank you for your attention.